Hello, I'm Essay, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
The turn of the 21st century witnessed a scientific drama of unparalleled scale and consequence, a narrative culminating in the near-simultaneous publication in February 2001 of two draft sequences of the human genome. One, appearing in the journal Nature, was the product of the publicly funded International Human Genome Project, a consortium representing the methodical, collaborative spirit of traditional academic inquiry. The other, published in Science, emanated from a private corporation, Celera Genomics, and was driven by the singular ambition and iconoclastic vision of its founder, J. Craig Venter. The 2001 paper bearing Venter's name, The Sequence of the Human Genome, is a landmark document, yet its true significance transcends the 3 billion base pairs it describes. It represents the apex of a methodological insurrection, a catalyst that irrevocably altered the pace, economics, and culture of biological science. An analysis of this work reveals a legacy defined as much by its competitive provocation and its transformation of scientific practice as by the raw genetic data it presented to the world. The intellectual landscape into which Venter launched his audacious enterprise was dominated by a single, colossal undertaking. The Human Genome Project, or HGP, initiated formally in 1990, was conceived in the grand tradition of 20th century big science projects like the Manhattan Project or the Apollo program. Its strategy was cautious, systematic, and hierarchical. The chosen methodology, known as hierarchical shotgun sequencing, involved a painstaking preliminary step, the creation of a comprehensive physical map of the human chromosomes. Researchers would cleave the genome into large, manageable fragments of approximately 150,000 base pairs, clone them in bacterial artificial chromosomes, and then meticulously order these clones along the chromosome to create a scaffold. Only after a fragment's precise location was known would it be shattered further for sequencing. This map first, sequence second approach guaranteed a high-quality, contiguous final product, but its progress was deliberate and immensely expensive, a testament to the prevailing belief that the human genome's complexity demanded such a methodical, almost architectural approach. Craig Venter's career, however, was built upon a foundation of challenging such orthodoxies. While at the National Institutes of Health, he had already demonstrated a preference for speed and massive data acquisition with his development of expressed sequence tags, or ASDs, a technique for rapidly identifying active genes. This work signaled a philosophical departure from the gene-by-gene focus of classical molecular biology, favoring a high-throughput, data-intensive perspective. The definitive proof of concept for his revolutionary alternative came in 1995 with the successful sequencing of the first free, living organism, the bacterium Haemophilus influenzae. For this, Venter employed a strategy he termed whole-genome shotgun sequencing. This method circumvented the laborious mapping stage entirely. It proposed to shred the entire genome into a confetti of small, random, overlapping fragments, sequence them all in a massively parallel fashion, and then rely on immense computational power to find the overlaps and reassemble the full sequence from scratch. To many in the HGP, applying this to the vastly larger and more repetitive human genome seemed reckless, a computational nightmare akin to reassembling a library of encyclopedias from shredded pages collected after a hurricane. Venter's success with Haemophilus influenzae, however, was an undeniable demonstration of the method's power and a direct challenge to the HGP's established dogma. It was this success that emboldened him to found Celera Genomics in 1998, with a singular, provocative goal to sequence the human genome faster and cheaper than the public effort. The culmination of this endeavor, the 2001 Science paper, detailed the application of this audacious strategy on an industrial scale. Venter's team combined cutting-edge, automated capillary sequencers with groundbreaking computational algorithms, the latter developed under the leadership of bioinformatician Gene Myers. The core innovation was the synthesis of high-throughput wet lab automation and sophisticated software capable of navigating the labyrinthine puzzle of sequence assembly. The paper presented a draft sequence covering over 90% of the human genome, assembled from 5 distinct individuals to create a composite picture of human genetic information. Its findings were profound and, in some cases, humbling. The analysis suggested a far smaller number of protein-coding genes, approximately 30,000 to 40,000, than the previously estimated 100,000 or more. This revelation delivered a subtle but potent intellectual shock, dismantling the simplistic notion that an organism's complexity correlates directly with its gene count. It redirected scientific attention toward the complexities of gene regulation, alternative splicing, and the vast, supposedly silent expanses of the non-coding genome. The paper also provided an unprecedented catalogue of genetic variation, primarily in the form of single nucleotide polymorphisms, or SNP, laying the essential groundwork for the future study of human diversity and disease susceptibility. The immediate impact of Venter's publication, in concert with the HEPs, was a global media event, symbolized by the carefully orchestrated White House press conference featuring President Bill Clinton standing between Venter and Francis Collins, the HEP's director. This image of unity, however, masked a deep and often acrimonious rivalry. Reception within the scientific community was complex and deeply divided. Venter's achievement in speed and technological execution was undeniable. He had forced the public project to accelerate its own timeline dramatically, arguably delivering the genome to humanity years ahead of schedule. Yet, the work faced substantive critiques. A central point of contention was Celera's initial business model, which proposed to make the sequence available through a subscription service, with significant restrictions on data redistribution. This clashed directly with the HEP's commitment to immediate, unrestricted public data release, a principle enshrined in the 1996 Bermuda Principles. The controversy ignited a fierce debate about the ownership and commercialization of fundamental biological knowledge, a debate that continues to resonate. Furthermore, critics pointed out that Celera's assembly, while rapid, was a composite draft that relied covertly on the HEP's publicly available mapping data to assist in scaffolding its own fragments. This crucial detail complicates any narrative of a pure, independent victory and reveals a symbiotic, if fraught, relationship between the two projects. The public project's sequence was arguably more complete and contiguous at the moment of publication, a testament to the quality control inherent in its slower methodology. Despite these controversies, the enduring legacy of Venter's 2001 effort is secured by the ultimate triumph of his core methodology, whole-genome shotgun sequencing. Once dismissed as computationally infeasible for large genomes, became the undisputed gold standard for all subsequent sequencing projects. Its efficiency and declining cost democratized genomics, making it possible for individual labs, and eventually commercial enterprises, to sequence genomes of staggering complexity. The race to the finish line, catalyzed by Venter's challenge, threw open the gates to the post-genomic era. The availability of a reference sequence powered the development of entirely new fields. Pharmacogenomics began to explore how an individual's genetic makeup affects their response to drugs. Population genetics was revolutionized by large-scale initiatives like the 1000 Genomes Project and the UK Biobank, which built directly upon the technological and analytical foundation laid in 2001. Venter himself continued to push the boundaries, sequencing his own diploid genome to provide a new level of personal genetic detail, and later pioneering the field of metagenomics with his global ocean sampling expedition, which used shotgun sequencing to characterize the genetic diversity of entire microbial ecosystems. The influence is direct and traceable. The analytical tools and high-throughput ethos championed by Celera became the bedrock of modern biological data science. In conclusion, the 2001 Celera Genomics paper is a document of profound historical and scientific importance, though its legacy is irreducible to a simple narrative of victory. It was a monumental technical achievement, a demonstration of how industrial-scale automation and computational prowess could solve one of biology's grandest challenges. The findings within the paper reshaped fundamental concepts of human genetic complexity, but the work's most lasting impact lies in its role as a disruptive agent. Craig Venter's private endeavor, fueled by venture capital and competitive fire, held a mirror to the world of public science, forcing a painful but necessary re-evaluation of its methods, its pace, and its relationship with commerce. The fierce competition, while often generating more heat than light, ultimately served the public good by drastically accelerating the completion of a foundational resource for all of humanity. The dual publications of 2001 represent less a finish line with a clear winner, and more the firing of a starting pistol for a new kind of biology, faster, more data-intensive, and forever intertwined with the complex forces of innovation, competition, and the perennial question of who owns the code of life itself. The sequence it described was a draft of the human genome. The process by which it was obtained became the final draft for the future of genomics.
We really appreciate you tuning in to this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.